Let me talk about DNA profiles, right? Um, yeah, this talk is going to be based not on the research that I have been doing, over the years. My research has been in classical fungal genetics, neurospora genetics. That I'm not going to talk about that, but I'm going to talk about something that I've been associated with ever since I went to CDFD in 2012, and that's CDFD's DNA profiling services. And it will be more a reflection on what I have observed in the DNA profiling services. And the main take home message for me, by the way, for people who haven't found a seat, those things on the side are also chairs, right, in the panel. So people can come sit on that as well. So the main point that came across to me is that obtaining DNA profiles is not very difficult, right? It's now standard, and in CDFD itself, hundreds of profiles have been obtained over the years, and that's not the issue. It is sometimes you need to know just that much more genetics to understand the significance of some of the things that are seen, right? And not knowing that much genetics, right, is sometimes comes in the way of doing full justice to the message that DNA profiles can give you. So in a sense, this is a bit of the applied genetics, you might say, right? We are talking about doing this mini-symposium is titled Genetics Basic and Applied, and this is the applied, and this is a widespread misapprehension about what the ethical problems can be. But I'll first describe. Oops. So this is just to give you an introduction to what DNA is, because I know there are a bunch of students here. On this thing, you will see there's what we call a backbone. Yeah, there is this backbone consists of these molecules called sugars, and there are phosphates attached to these sugars. So there's this sugar on this 5' carbon, right? There is a phosphate, and also on the 3' carbon here there is another phosphate, and then this phosphate is connected to the 5' of this sugar, and this sugar is connected to the on this 3' to another phosphate, and this goes, all along. There is a ladder. Okay, I thought that people on the webcast may not be able to see it. Sorry. Yeah, this is to go. So these are the sugar phosphate backbones. One backbone goes 5' to 3' on this direction, and the other one goes 5' from here to 3'. So they are called antiparallel strands. And then attached to each of the sugars is a base. The base can be either this double ringed structure called adenine, or it can be this single ring structure called thymine, or it can be this single ring structure called cytosine, and this double ring structure called guanine, right? These are the 4 bases. So in DNA, adenine is always paired with thymine, and cytosine with guanine, right? So if you have the sequence going 5' to 3' of adenine, cytosine, thymine, and guanine, then you can predict that the sequence on the other strand going 5' to 3' is cytosine, adenine, guanine, and thymine, right? And this is just 4 bases shown in the DNA. Our DNA in our cells is 3000 megabases, right? So that's 3 billion base pairs. So that's a lot of DNA. And in this DNA, there are some interesting sequences which are called simple tandem repeats. So basically, here's consider this DNA sequence, here. From here to here, you have a sequence of G, C, T, and so on and so forth. And but then, and again it resumes, there you have C, G, the sequence goes on. But in this middle stretch, there's CTT, 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 repeats of a simple sequence. It's a 3 nucleotide sequence that is repeated. In this case, 8 times, right? Now in certain other individuals, because these repeats are susceptible to slippage and the number can change during replication because of that, in an other individual, the flanking sequence would be the same, but instead of having 8 repeats, you could have 7 repeats, or instead of having either 8 or 7, you could have 9 repeats, right? Now this is a very interesting phenomenon, because using the same oligonucleotide primers in a PCR reaction, and I am assuming, since many of you students are from genetics and biotechnology, that you know what a PCR reaction is, you can actually use the same primers to anneal to these outside flanking regions. But if you do PCR from here, you will get the shortest allele, because it has only 7 repeats. From here you will get the longest allele, because it has 9 repeats, and here you will get intermediate size allele, because it has 8 repeats, right? So if you run this in a very simple setup here, this is what many of us used in simple experiments. This is a agarose gel electrophoresis. You can separate the amplified DNA based on size, right? So in each individual, you will see that you get 2 versions, a longer and a shorter one, depending on the size. And if they are of the same size, you get one big thick band of a single size. So it's very easy to score this. Now it turns out that the human genome has more than 700,000 STR loci. Okay, there are STR loci on all the chromosomes, and there are more than 700,000, approaching 1 million, right? And at all of these STR loci, you see considerable polymorphism. In fact, now that about 1 million human genomes have been sequenced, it is possible to look at the relative polymorphisms in different STR loci, right? And despite this very vast amount of possibilities, we focus on only 16 of them, right? Or for the most part, only 16 of them, because they were chosen by the FBI many years ago for DNA profiling studies, right? So those 16 are shown here. The way these amplifications are done is in a multiplex PCR. The way a multiplex PCR works is that, we have on the primers for the oligonucleotides, we put a fluorescence detector, right? So a fluorescent moiety, and the primers are so designed that for this locus, which is amelogenin, you have the range of sizes is here. For this particular locus, this is all the possible range of sizes. For this one, this is all the possible. So for these 3 loci, if you use the same red fluorogen, right, you can separate the 3 loci. You won't have an FGA allele coming and overlapping here, or this allele coming and overlapping there. And likewise, you have 4 different fluorescent sort of signals that you can pick up, and these 16 loci are represented here. So that's 4, 4, 5, and 3. 16 loci from all over the genome, and this is the range of alleles. So by doing a multiplex PCR and then running it out on a capillary column, so the thing is a bit different from what I showed you in the previous slide, where it was a simple slab gel, right? We can actually get sharp peaks for the 2 alleles, right? So for this particular locus, one allele has got this 13 mer peak and the other has got the 14 mer peak. And likewise for various things, whereas here for one single, this could be the second allele, but it's quite possible that this is a double peak, right? Whereas this could be a stutter peak or a background peak. Now, there are standard ways of determining whether it is whether this is a real peak or not, right? So once you get this profile, right, we can put it into a table. And this is actually taken from an actual case that was handled at the CCM. This was a technical examiner, this was the director at the time who, when this case came up, and this is a stamp and it's all official, right? So what was done here is that there was a crime scene from which the police had got a sort of spot of soil on which there was spit, right? And there was also a knife found at the crime scene, Yeah, thanks. There was a knife found at the crime scene where we got the DNA from that as well. So DNA profiles were made from the DNA obtained from the knife handle, from the soil in the crime scene, and from one suspect. I blanked out the suspect's name because I didn't want to, it's the proper thing to do. Now you can see that the suspect has 2 alleles at each of the 16 loci, and the soil that was found at the crime scene also has the same alleles. So it's almost like a CCTV camera has picked up that this guy must have been there, right? So the police are now pretty confident, and also from the knife they got many alleles that are present in him, although they also got some alleles that are not present in him, which means that the knife could have been handled by more than one person. And when you do the PCR and so on, you can pick up alleles from the other people who have handled it as well, right? So this is the power of DNA profiling. If I had to give one quick example, here's how we can show that this, for all our purposes, this proves that this guy was at the crime scene and he spat there. He'll have to explain how his DNA happens to be there, right? So this is one standard triumph, and we can get DNA, from people's clothes, towels, switches, chewing gum. I think some of you might remember that there was a time when Pranab Mukherjee was a finance minister, right? Somebody had found a microphone under his desk stuck with chewing gum. There was a big furore in the news. They sent the chewing gum to CDFD examiners, got the profile, and then the police didn't want to do anything. They didn't ask us for the profile. They just pretended that everyone's forgotten. Time had gone, right? But we still have the profile, and we can't do anything with it unless the agency that sent it to us comes and claims it. And if they claim it, they'll be able to find who chewed the chewing gum, right? So they rather not deal with some of these issues. So, but we have the profiles, right? Now I'm going to tell you about some getting into the ethical angle. One of this is a crime, again handled in CDFD. This is a crime of incest, where a father raped his own daughter and there was a baby produced, right? So this shows you, this is the father of the victim, this is the victim, this is the father's wife, and you can look in the DNA profile of the victim and you will find one allele coming from the father and one from the mother, right? That you will find. So this shows that she is the product of this mating. But when you look at the baby of the victim also, that's this one, you find that the parents of this baby are this man and this daughter, right? So this proved, right, that this was a case of incest. And what I did after I saw this profile is I just highlighted with a highlighter all the loci that are homozygous, which means in this baby of the victim, this particular locus has the same allele, 11, 11, 8, 8, at 4 loci. Now, this means that homozygosity is become homozygous at 4 loci. 4 out of 16 is 1 by 4. This is what you would expect in a first degree mating, right? So, and this I'll show you in the why it comes to 1 by 4. That is, if you have, 15. Okay, that's pretty good. So I'm doing well on time. So if you have a father-daughter mating, the probability of the locus being homozygous is 1 by 4. That's because if you consider one allele coming from the father, let's say that goes from with the probability of 1, the other allele could be the same because that could be inherited by the daughter, right, with a probability of half, because the father has 2 alleles. He can either pass on the allele he's passed on here to his own daughter, or he can pass on the other one. And then she has 2 alleles, and there's a probability half that she'll pass on the same allele down to this individual. So the probability that the same locus can be homozygous is half times half, which is 1 by 4. it translates to the fraction of the genome which is homozygous by descent, F, right? And this is the same for a brother-sister mating as well. And in an uncle-niece mating, this man is a brother of this lady whose daughter he is married. And these marriages are actually quite common in certain communities in India, in Andhra Pradesh and so on. So in an uncle-niece marriage, F is 1 by 8, right? And then there's first cousin marriage, and there's, there can be further relatives and so on. So there is this F parameter, which is called the coefficient of inbreeding, gives the fraction of the genome that is homozygous by descent. And I told you that for a father-daughter mating, it is 1 by 4. And looking back in the baby, 4 out of 8 loci are homozygous, which is 1 by 4, right? Consistent. But when I did the highlighting, I didn't just highlight in this baby who is the product of a father-daughter mating. I also did it in the victim and the father and his wife, right? And in 2 of them, again we find there are 4 loci that are homozygous by descent. Does this mean that these individuals are also produced by a first-degree marriage? No, it does not necessarily mean that. Right? It could, but it does not necessarily mean. There is a possibility of getting 1 by 4 of the genome homozygous by descent by means other than a first degree mating. Right? And in India, it's fairly commonly seen. This is known as endogamy. Endogamy is when marriages occur within the same community. Right? So, while this result is consistent with first degree mating, these results could be due to endogamy. The fact that this was a case of incest is not in doubt. It's based not on the fraction that is homozygous, it's based on the fact that the baby had 1 allele from this victim and 1 allele from the father, right, from her grandfather. So that's what shows that it's incest. So I've talked to you now about several things, right? I've talked to you about basically what DNA is found in every cell, which is why when the person spat it or when he handled a knife, his DNA was left there. I've talked to you about how profiles are made using the STR loci, that there are large numbers of STR loci, but we only use 16 of them because they are the ones that the FBI has standardized and everyone standardly uses it. Using some other 25 wouldn't make sense because then we can compare, right, across things. And I've shown you how F, being 1 by 4 is satisfied here, but it is also shown here, which does not necessarily mean incest. So things can be complicated. You can get F 1 by 4 even though it is not a case of incest. Now this is a little bit subtle, right? And this is where the ethical issues come up. Actually, the champion DNA profile that I found in CDFD, right, was this one. Here was a case of paternity, right? There's Mrs. So-and-so and Mr. So-and-so, and this Mrs. So-and-so accused, from what I can remember, this person as having fathered the baby. And then we did the profiles, DNA profiles of the trio, and found that that was not the case because there is these alleles which are marked out in red, couldn't have come from the father and they couldn't have come from the mother, right? So the alleles in red excluded this person as the father. Now, as far as the DNA work is concerned, this much is enough. We could have just sent this report away. But I have told you that when you are a geneticist, you get interested in seeing how many loci are homozygous. And when I looked at this profile, I could see that 50% of the loci are homozygous, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, more than 50%. And I told you that 1/4 is in a father-daughter mating, right? So this was strange. She probably knows who the actual father is, right? She has accused him, but we can show is not the actual father. But she is not accused the real father, right? But we can see that there is so much homozygosity. what should we do, right? In such a situation, is she scared of implicating someone who could be a first degree relative, right? And in writing our report and telling the police this, just what we have told them, are we actually evading the responsibility that it could be a first degree relative, right? Or is it possible that this population is so endogamous that you could have 50% of the genome homozygous by descent, right? I'm going to not tell you what we did, right? Because if I tell you what we did, then we go off into that I said this and you said that. I want you to ask yourself, what would you do in such a situation, right? You guys are the DNA examiners. The police has come to you with this case, and the police has now asked you for the report. Simplest thing is to exclude him, and that's it. You as a geneticist, right, have seen this. Are you going to keep quiet? Are you going to do something about it? But as a geneticist, you know it can also be because of endogamy. Are you going to accuse someone of incest when it could be endogamy, right? This is the ethical question. This is a question that DNA examiners, right, are not trained to deal with. This is not something that even I as a geneticist, and I have spent many years doing classical genetics, even I have not been trained to deal with it. I want you guys to ask yourself, what would you do in such a situation? Is it possible that by taking the easy way out, just reporting exclusion, we are actually guilty of knowing that there might be a crime happening here and are withholding this information? I'd really like to know, what people who deal with this, activists who deal with this think, right? Like women's groups and judges, police, lawyers, other people who have thought about this. I'm going to just leave it at that. I'm not going to answer what we did. Oh, this is the last. Okay. So basically, the question I stopped at was whether there is a possibility of incest. So, in short, there is one other slide. This slide, if there is time, how much time do I have? About 3 minutes? 3 minutes. Okay. So this slide shows you another case where simple things are not understood by very important people, right? This is showing you the landslide in Uttarakhand in June 2013. This is a picture taken, I think, of the devastation in Rishikesh. This statue eventually toppled, right? It was estimated that 4,000 people went missing in that landslide. Actually, now the number has been revised up to about 6,000. There were 600 mortal remains recovered in that year. People went from CDFD, actually were lowered by rope ladders, because they were doing the cremations in situ. And before the body was cremated, they actually would take bone. Most people don't know which bone to take. Suppose you find a dead body and you want to get this DNA, which bone will you get? Bones are not easy to get, by the way. Yeah, you need to know how to remove a bone from a body from which you can get DNA. Anyway, there were 600 mortal remains, and at that time, when this slide was made, 400 remains were profiled. Since then, all 600 remains have been profiled. 200, we advised, the CDFD advised the Uttarakhand government that try to get DNA from relatives of the missing persons. And they were able to get of about 200 relatives who had come to Dehradun and left their DNA behind. But we had told them that these are people coming from all over India, right? And there'll be people from Karnataka and Telangana and so on and so forth. And you should find a way of getting them to send their DNA of these 4,000 to 6,000 people to CDFD. But we got only 200 relative samples, and we profiled those, and we found 18 matches as of July 2014. In July 2014, I went to Dehradun, right, to meet with the additional chief secretary, and on the anniversary of this event. And somebody asked, should we make a press release? He said, "Arey, what's a press release? We only got 20 matches." So I had to. It took me a few seconds. I thought either stupid people can become principal secretary, chief secretaries of a state, or when somebody becomes a principal chief secretary of a state, he can become stupid and he thinks he'll get away with it. But one of these two it is, right? Because we told them that about 4,000 to 6,000 people are missing. About 600 remains have been found. That's 10%. You got 200 relatives. 18 matches were found. That's 10%. Do you expect anything bigger than that, right? The main thing was they should have increased the number of relative profiles. And that could have been done if they had only announced in the papers and the news media and so on that anyone who has lost a relative send a drop of blood on a blotting paper to CDFD. We'll get the profile made. We'll then match with all the remains that we have and we'll know whether that person's relative was missing. That wasn't done, right? So this number, which they thought was hardly worth reporting because it was thought to be a failure, that these DNA profiling people failed us. They only made 18 matches, right? That is another misconception. It would have worked had we got more of those remains. We don't have the bandwidth to get the DNA, but the National Disaster Relief forces and so on can easily advertise. It can still be done, by the way. If anybody in this audience, right, has a relative who was missing, first degree, like son, daughter, brother, sister, etc., they send us the DNA, we can still do the profile in CDFD, right? And we can determine whether the person's loss was there. So I think with that I'm going to stop, right? My talk is over.